then we have to analyze that phenotype in some detail. So we only know what we can see experimentally in the organism, so we need some appropriate markers, some appropriate microscopes, and we need to really look at this phenotype in detail and appreciate what's going on here, even if it's subtle. You know, one cell bigger than the other, that's kind of subtle, but it turns out to be important. Then, based on what goes wrong in the mutant, we would infer what should have happened in the wild type. And so we begin to build up a story of this is what should happen in the wild type case with this timing and this spatial configuration. Ultimately, we do have to figure out the gene that was mutated to create that phenotype and think about what is that gene doing in a biochemical sense. So sometimes, on exams, I'll ask a question like, what's the biochemical function of this gene? And by that I mean, is it a transcription factor? Is it a cell receptor? What is this protein product actually doing within the cell? Okay. And then this can just spur a lot of additional questions about how come a mutation in that particular gene would cause that particular phenotype. And the more that we understand this reason, the more that we understand the internal natural workings of the cell, or the mechanisms of that cell as it is accomplishing this task, such as how is the cell accomplishing differentiation into a body muscle cell fate. Back in lecture 1, I think it was, I said that if you discover a new gene that is critical for some process, one of the basic kinds of follow-up questions that you would do is ask exactly when and where is the mRNA made, or exactly when and where is the protein made for that gene. So the Confuse lab, after they found these PAR mutants and they cloned the gene, one of their next questions was to make primary antibodies for each of these and to do immunohistochemistry and find out where is that protein localized within the embryo. And here is the result for 2 of these proteins, the PAR3 protein shown in green and the PAR2 protein shown in red. This is a one-celled embryo shortly after fertilization, and look at what they found. It's really unusual. What we see is PAR3 protein is localized right at the cortex or periphery of that cell, but only on half of the cell. Okay, PAR2 is also at the cortex on the other half of the cell, and I can't even really imagine what these proteins are doing that they would need to have this localization. So sometimes you can also get a clue to protein function based on a blast search. So in your first lab, you found a protein, you did a blast search. You're basically asking, out of all the proteins known to mankind, what is similar based on sequence to this new protein that I found here? And you can find some information about gene families or particular conserved domains that could give you some hints as to what this protein is probably doing biochemically within the cell. Okay, so for example, PAR1, we found out it's an enzyme, it's a serine threonine kinase, so that's probably going to be something that's involved in communication or signal transduction from surface to the center. In the case of PAR3, it was totally novel, so there was nothing out there in any of the databases that gave us any clues at all. It's good and bad if you're a graduate student, right? You get this interesting new gene, but you have no idea where to start with it. PAR2 is a zinc binding protein. Sometimes that can be transcription factors. So, C. elegans, as we said, these oocytes, as they come down the pipeline, are encased in an eggshell. And as they come down the pipeline, the egg nucleus is sort of at the trailing end of this oocyte. The sperm are typically going to fertilize through the eggshell on the opposite side. And so here is the sperm pronucleus that has just been deposited in this egg when sperm membrane and egg membrane fuse. The sperm are also bringing in a centrosome. So a centrosome, you'll remember, is a microtubule organizing center. And all those red lines in this diagram are meant to diagram microtubules. And so in C. elegans, it happens that the site of sperm entry is consistent, and this is going to provide information to the embryo to establish the anterior, posterior axes. So in frog, it was the information for dorsal ventral axes, but in this organism, slightly different. It's going to be anterior, posterior, set up immediately by the site of sperm fusion. So as these two nuclei are now coexisting in this egg, they're going to need to migrate together and fuse to recreate that diploid nucleus. And as they do so, they're extending microtubules from the nuclei to the cortex of the egg. And as time goes by, these nuclei migrate towards the middle and do meet up and fuse. And meanwhile, they've grown out these microtubule arrays reaching from the nucleus all the way up to the cytoplasm. And on the one side there, the right-hand side, we have those microtubules associated with PAR2 protein. And on the other side of the egg, the microtubules associated with PAR3 protein, both at the cortex. And this whole mechanism that's being set up seems to influence exactly where the metaphase plate will form during that first mitotic division. So this will be where the chromosomes align together prior to the two daughter cells separating in anaphase and telophase. And this will also determine where the cleavage furrow forms for that embryo. That is how exactly that embryo is going to divide into two daughter cells and how big and what cytoplasm and so forth. And so eventually then we get down to the two daughter cells, and this unique pattern of PAR expression or localization within the embryo continues. So C. elegans can teach us a lot about some of these very early decisions to establish a particular commitment. So let's look in a little bit more detail at how that works. So within this window of choosing or becoming committed, we have two processes that are segueing into each other. The first initial process is specification, and this is, we've touched on this briefly before, this is a period of time where cells acquire enough information to start to differentiate into one or another cell fate. So if they're just left alone in kind of a neutral environment, at this point they're beginning to have enough information to go ahead and differentiate a particular cell fate, but they could still change that cell fate if moved to a new place or exposed to different factors. They're still malleable or changeable. And then later in time they become a lot more fixed. So at this point they're actually really committed to form one particular cell fate, and are not subject to being changed even if exposed to external factors. So we could look at a couple of different ways this could play out for cells within embryos. If we think about the initial acquisition of information, this could happen in a cell-autonomous fashion. This is the case for most invertebrates, such as the sea urchin, C. elegans, Drosophila, are going to use this sort of a mode. And what this means is that the cell is mainly relying on itself, its own intrinsic cytoplasm and genome, to make the decisions that it needs to choose one cell fate. So all of the mechanisms that influence that choice are within the cell itself. It's not really asking or interacting with neighboring cells. It's not really sampling its local environment within the embryo. It's simply relying on maybe its lineage and on some kind of cell intrinsic types of information. So if this is the case, then that means that particular cells are going to be very fixed in what kind of fate they adopt from one individual to the next. And we saw that, in fact, that is a feature for C. elegans, that each cell is very fixed as time goes by. So we can think about what is the experimental evidence that tells us that this mode of cell fate acquisition is at play for C. elegans. And so one way to do this would be then to take the cells, separate them from their neighbors so there's no possible way they can interact with the neighbors, and see what they can do, see what they have in them in terms of dividing and continuing to give daughter cells. So if you separate the A, B cell and the T cell and then you culture them, so you're keeping them alive and happy, just ask what sort of progeny they can produce. And it turns out that this P1 cell can still make the exact same number of progeny that it would have otherwise. So that seems like a cell intrinsic characteristic. And those daughter cells, in general, give rise to the normal types of cell fates that you would typically see from P1 cells, so germline cells. And if you look at P granules and how they're segregating in the future daughter cells from this, it's fine. The PAR genes seem to be functioning fine. So that really does imply that it's mainly cell intrinsic kinds of mechanisms at play here. So we can conclude then that, especially for the development of the germline, which is one of the PCL fates, that this is a cell intrinsic or a cell autonomous mechanism driving this process. And in general, C. elegans largely relies on cell autonomous mechanisms for separating cell fate. Sort of a well-known characteristic for C. elegans. So here's just another example of this, just a different marine invertebrate creature known as patella. And one of the cell fates is that some of these cells in this organism, as it develops, will develop these cilia that swish around some of the seawater and maybe bring nutrients closer to it. And you can find cells in the early embryo that are beginning to be fated or specified to be these trophoblast cells. You can separate them out and isolate them, culture them individually, and once again you get the normal number and the normal cell fate from these individual cells, indicating the extremely cell autonomous mechanism driving them to take on this particular fate in life. Okay. So when you have this sort of scenario where many of the cells are relying on intrinsic or lineage-based information to take on cell fates, we can put that together to a general mode or pattern of development that we call mosaic development. And the analogy that's often used to try to talk about this is the tile mosaic. And so here's a tile mosaic, and what you can see is we have a number of individual tiles, each one of which is a pure color, or single cell fate. And they're organized in a particular arrangement. And in fact, it is this arrangement next to each other that gives some kind of higher order magnitude of organization to this tile mosaic. So we could have just taken the same exact number of tiles with these colors and put them together in any number of different patterns. But when you put them together this way, you get a flower, you get some leafy structures, you get a repeating type of border. So you get some function or some organization and some added meaning to this just based on how the tiles are arranged. And if you were to take a hammer and break some of that and think about what you would need to replace that using what's available to you, here we have blue cells and white cells, and if those cells were, say, able to divide, you could imagine them making more blue or white cells, but it's going to be hard for them to make a green cell because that's something totally different than what they were originally. So in the case of tile mosaics, if you have somehow removed or injured cells early on, there's not a great ability of the neighbors to compensate for that problem and regenerate the missing cell fates. So often you'll just have kind of a blank or absence of those cell fates within the embryo. So in C. elegans, if you take a laser and fry one of the daughter cells, such as the P4 cell, the other cells keep going and they make their normal number of progeny, but then that P4 cell is never replaced by the neighbors, and you lack a germline for that embryo. Okay, so mosaic development and cell autonomous lineage are two related concepts that go together frequently. Okay, and part of the reason for this is because the way that cleavage is happening here, because it is the same in every individual, you are in fact dividing that cytoplasm in a very specific way. And so it does make sense that lineage could be important here and that each cell might be inheriting particular factors. So let's look at the opposite possibility. This conditional specification is the other possibility here, that you're going to see more in vertebrate organisms, but a few invertebrates as well. And in this case, instead of relying on lineage, you're going to rely more on cell interactions. So there'll be a lot of communication between these cells, crosstalk back and forth, and based on that, you will then make the ultimate decision about the cell fate. And you often see this sort of case where the cleavage pattern in embryo, one individual to the next, is not exactly set, can be a little bit variable. And then any particular daughter cell, if you point to it at that particular time from one individual to the next, it could be within maybe a smaller range of possibilities, but still more variable, more flexible. So just as an example of this, there are a few cases of C. elegans, a very limited number, where we can see that some degree of conditional specification is at play. And so in this case, we're talking about the AB cell cultured alone now. You actually get fewer progeny, and you're actually missing some cell fate, such as, for example, anterior pharynx fate. So this particular cell actually did need to interact with the P1 cell to some degree in order to generate the full range of daughter cell fates, including number and type. And so they're not completely cell autonomous, and cell interactions must be important. And here's kind of a summary of some of these concepts put together so far. So in C. elegans, we see mainly mosaic development, where we can't really compensate. And the reason for that is because we have generally a mode of autonomous cell specification. And here's some of the evidence for this, that you can find isolated P1 cells that still behave perfectly normally, that you can actually see some of the factors that might be responsible for these lineage-induced fates, but that it's not a perfect 100% autonomous specification. You can find a little bit of conditional specification too. Okay, I think I have maybe one or two more slides, and then we'll take a break. So, if you have, on the other hand, extensive conditional specification, then we're going to go to a mode called regulative development, which means that the cells will be able to compensate, divide, and reestablish a limited number of fates if some of them are missing. So here, for example, is a frog embryo. We've just taken this needle and swapped out a few cells to create a missing chunk. Now, if this were a tile mosaic, that would be it. It would never be replaced. But in the case of the frog, these neighboring cells somehow sense this. They continue to divide, they reorder, rework it out between themselves, have a discussion, and then they are able to fully reestablish those missing fates, and now you get a normal embryo that develops. Also, if you think about this experiment, you start out with a group of cells that are in a certain position in an embryo. If this were cell lineage-based or cell intrinsic mechanism, if you were to move those cells to a new location, you would not change their fate. They would still always be eye cells or ear cells, depending on what they were originally. But with regulative development, since there's so much interaction and like feeling your way, talking to your neighbors, working it out together kind of thing, if you do this early and you move these cells to a new location, yeah, they can change their fate and will adopt the location based on the neighbors that they have and the new position that they have, and become an entirely different piece of body in the adult or larval organism compared to what originally they ordinarily would have become. So regulative development is more common for vertebrates, but you can see the evidence of this in invertebrates as well. So here's a sea urchin where it's been separated out at the 4-cell stage, and you get 4 pretty decent looking embryos out of this. They all have all 3 germ layers. They have all the cell fates involved. Their morphology is a little funky, which is hard to know if that was experimental induced or it's really reflecting the capacity of these daughter cells, but it is some evidence of regulative development. And then the final slide here with humans. Monozygotic identical twins. This is a situation that can happen when you get one sperm fertilizing one egg within the mother. That zygote then divides, and somewhere during the early cleavage period, this has to be pretty early, this thing separates into two different bundles of cells. Well, each of those bundles of cells can regulate, they can reestablish themselves as a complete and separate individual and give rise to two offspring. And here are two identical twins, which I just think this is just amazing how similar they look to each other, which they probably enhanced by, wearing the same top and so forth. But it does kind of show like the extent to which regulative development could really operate here. That even though you have fewer of the original cells, that means they're both totally normal individuals here. Okay, any questions on this? Okay, let's take a little break here. It's 8:54 now, so let's come back at 9:04.